moving on to our next speaker is Justin Montgomery, who is an associate research fellow at Pfizer. Justin joined Pfizer Medicinal Chemistry in 2006, and he received his Ph.D. from University of Illinois, working with Sept, Denmark. And while he's been at Pfizer, he's worked on a variety of things, as you can see from his bio, including HTS triaging, fragment-based drug discovery, protein degradation, a little bit of everything. He's moved more recently work focused on DEL hit triaging, which is what he's going to be talking to us about today and how they do things at Pfizer.
Thanks so much, Lisa, for the introduction. Thanks to all the organizers for inviting me to present at this wonderful forum so far. A big thanks to Paul, who just presented, because you'll see a lot of similarities in the talk that I'm going to talk about today, although we got to kind of the same place using different routes. So very interesting. So, of course, our overarching goal is to discover medicines at Pfizer that improve people's lives. And so the hits that we derive from our DEL screening really need to be attractive, and we need to be able to optimize those into drugs. And when we started the whole DEL process at Pfizer about 5 years ago now, the hits that were coming out of the screens were generally very high molecular weight, high lipophilicity, and project teams were unable to optimize those hits to workable leads. So we took a two-prong approach to get to a better place, and that's what I'll talk about today. The first part of that was improving the properties of our libraries, and I'll touch on that in the next slide. But the other part was really making properties front and center during analysis, and developing an analysis tool that allowed for flexible triage of these huge DEL data sets. So, of course, our goal is to not only find, but also prioritize the hits that have the best combination of not only high reads, but also that great primary SAR that's in the data, which is very useful, attractiveness, which includes good properties, minimal liabilities, and coverage of those multiple biological profiles, which is, as Scott described, the way a hit profile changes over the different selection conditions, so the biology that you're seeing there. And our strategy along the way here is always to just use as much of the data as possible and look at it in as many ways as possible, and not really silo ourselves into a certain process. So we really want something that's flexible here. And to do this analysis, you need some expertise in biology, especially chemistry, medchem, properties, data analysis, and of course this flexible analysis tool that I'll be telling you about, which is based in Spotfire. So today's talk is going to cover a quick introduction to our libraries, a little bit on properties and our DEL NPO score, and I'll give a very high-level view of our hit confirmation process, and then we'll go into a detailed look at the Spotfire-based analysis tool called DEL-DV, and then some insights on things like bead binders, frequent hitters, bioprofiles, all things that Scott touched on as well, or Paul touched on as well. And then one slide on how we use the tool just at a very high level to triage a screen, and then some final thoughts about what we do with hits after they're in our hands. So Pfizer's DNA-Encoded Libraries are number 33. We have 22 3-cycle libraries, 11 2-cycle libraries, and it's about 2.5 billion recipes combined, with 15 million of those coming from the 2-cycle libraries. We have 10 libraries that have an alternate DNA connection, and what I mean by that is not the standard amine DNA head group attached with a carboxylic acid to give you a methylamide when you cleave it off DNA. It's just something different from that. So this plot on the left is kind of showing the evolution of our early libraries, which are represented in the cluster in the upper right here, and our newer libraries in the lower left here. It's a plot of the average C-log-P and molecular weight for these libraries. And you can see the early libraries were kind of around 550 or so molecular weight, C-log-P 3 to 3.5. And this is an average of hundreds of millions of compounds. So of course, there's going to be molecules that are in that much better property space in these libraries, but also ones that are in that north of 600 molecular weight range and C-log-P of 5, where they start to get pretty ugly. And you can see how we reined in properties a little more in our later designs to get into that 4 to 450 range and better C-log-P range. But this was at the expense of library size, of course. And we currently screen all of these libraries. We haven't ruled out the early ones, but using this tool that I'll be talking about, we can pull out the good compounds from these early libraries as well. So since we're talking about properties, I want to introduce this multi-parameter score that we use. So an MPO score is just a way to combine multiple properties into a single score that then can be used for visualization or sorting purposes. We rarely use it as a cutoff, but we really take advantage of this as a to use for color when we're looking at all our cubes, to really draw your eye to features that may have better properties over other properties. So if you just use property cutoffs, you might get rid of compounds that are kind of close to the edges. It might be great for a few properties and just over the line on another, and you probably don't want to throw that compound out. So the MPO really serves to soften those edges. And when you're developing one of these scores, you really don't want to include every property you can, because so many things just track with molecular weight and polarity. So you really want to be make good choices when you're including when you're picking which properties to bring in here. So for our MPO, we use molecular weight, C log P, of course, and then we've brought in number of aromatic rings, which has an added benefit over the first two properties for predicting solubility. And it's very easy to calculate, of course, and hydrogen bond donor count, which gives you a little added with respect to permeability. And as we started adjusting these curves, which are shown here, so for example, molecular weight, we give it a full score of 1 up to a cutoff of 400, and a gradient down to 0, 600 and up is a 0, and you can see the scores for the other properties as well. As we were looking at all these structures and the scores that came out of them, and talking to lots of different medicinal chemists, it was very clear that molecular weight is by far the most important property here that makes things look ugly to medicinal chemists, myself included. So we wanted to weight that a little more heavily, and rather than applying a scalar to it, we ended up using a product for that particular score. So in the end, we take the molecular weight MPO score, which is a number between 0 and 1, multiply by that the sum of the other 3 scores, and that gives us a value between 0 and 3, and molecular weight greater than 600 is going to zero out that score, whatever the other properties are. We visualize this in our cubes in stoplight colors, and so you can clearly see line features that have compounds with better properties in it compared to others. So here I'm highlighting 2 individual compounds where the one with worse properties has higher reads. The MPO score is much lower, and you can see the molecular properties that went into the calculation of that MPO score. So you'll see these colors throughout the talk today, and it's all coming from this score. I do want to highlight that, a good MPO doesn't mean it's a good compound. It's never an excuse for looking at that structure and seeing if there's other liabilities in there that the score isn't picking up. But it's really a nice sorting tool that we found very useful. Okay, so our post-screen process has been described in a couple of papers here. So I'm just going to go through it at a very high level on this slide, and direct you to these 2 papers if you'd like to get some more details on the whole process. So we start with a feature analysis using this tool, DelDV, that I'm going to be going into great detail in the next few slides, which is basically our analysis tool for selecting things to take into the next step of the funnel. Instead of taking those right to off DNA synthesis, we like to go through a process of on DNA synthesis and then characterization on DNA. And this comes from the fact that we all know that these DNA codes are recipes for molecules, and it's not always what actually happened in the actual synthesis of the library. We talked about this with Chris in the XChem talk earlier on, how different validation yields can lead to different expectations for the yield of that targeted compound at the end of the day. We had pretty high validation yields when we made our libraries. I think it was 70% or so, but when you take that through 3 cycles of chemistry, there's certainly lots of things that can go wrong there. So we recapitulate the hit synthesis exactly the same way the original library synthesis was done, but as a singleton instead of in a pool. And then we analyze that through mass spec in order to try to identify the mixture of compounds that are in that mixture. And then we subject it to a selection experiment, very similar to the original DEL selection, where you can select with matrix alone or target containing matrix in an ASMS-like experiment. And that allows you to determine which component of that mixture is responsible for the actual target specific binding. In this example that comes from this paper on the right, a minor component was responsible for the target specific binding and not the targeted compound, which was present in higher yield. So this step really helps us determine what the real binders are, and if we actually recapitulate that binding in this experiment. I know there's been a lot of discussion on the usefulness of this platform, and a lot of times it comes down to people expecting to find these unexpected byproducts that have great properties that turn out to be the drug when you do this selection. But that's really not the value in my mind and to this platform. The value in this platform is weeding out a lot of stuff that either doesn't confirm in the on DNA step, or when it's a binder that is not your expected targeted compound, more often than not, it's a undesired binder. So it has some chemical liabilities in it. It's an unknown structure. And more often than not, the molecular weight is actually way higher than the targeted molecular weight. So the properties are getting worse there. So what this really serves for us is that it cuts down the numbers of things that we make off DNA, because we don't have a group dedicated to following up these DEL hits. We need to get the project teams to actually invest their own resource in following these up. So the more we can weed down that set of hits for them before it gets to them, the better. So shown here are just some numbers for the step that are kind of average for our process. Of course, we're starting from billions of compounds. We generally take forward less than 30, usually on the order of 10 or 15 or so to an on-DNA synthesis. It's usually one compound per feature. We always get the same answer when we do more than one compound per feature. And then, another decision point comes in by analyzing all of this on DNA data that really takes us down to less than 10 compounds per project, and often as little as 2 or 3 being made off DNA. And if that leads to a couple of tractable lead series, then that's a win for us. Oh, along this path, we do collect lots of statistics on library fidelity, all that stuff. I do have a slide that was approved for showing that I can, if someone wants to ask a question about that later, I'd be happy to flash that up and give you some real numbers about this process. Okay, so Paul introduced the cube very nicely. I just want to add a couple of points to that. So our cubes, like Nurix, are sorted by a chemical space, a similarity metric. So compounds that are close together are going to be, or monomers that are close together are going to be close together on the axis. And then the spheres, of course, are the individual unique DNA barcodes sized by the read count. Here I've highlighted one particular marker, and this can be decoded in this triazine library to these 3 building blocks around the triazine. Of course, we do a lot of the analysis around lines or planes in this data. All of the data shown in this cube is in a single plane corresponding to the red building block A, and everything in this line is corresponds to the red building block A and the blue building block C here. We actually characterize these line features in a couple of different ways. The first we call line feature normalized strength, and that's simply adding up all the signal in that line and then dividing by the number of monomers in that particular split in order to give us a overall strength of that line feature that can be normalized across differing monomer set sizes. And then we just take the same calculation and do a standard deviation calculation on it to get a line feature diversity, which is going to tell you whether or not this line is a uniform line like you see here, or if it's one of these features that's likely more interesting, where you just have a couple of things close in space and some are much better than others, where you might be seeing some real SAR. And like was mentioned, these line features can be explained in a number of different ways. The easiest to rationalize is that you're just seeing SAR for a particular R group. And when that's the case, that line feature is likely going to be a diverse feature, not one of these uniform type features. Another possibility when it is a uniform feature is that that particular R group is not important for binding. So if you imagine the magenta building block, which is variable in this line, is stuck out into solvent, you can put pretty much anything there and still maintain the same activity. You might see a line feature like this. But another possibility that actually happens a lot more than people think, I think, is that you're seeing these features due to failed chemistry, truncation, structure reactivity relationships, things like that. And these often are uniform lines as well. Just to illustrate that point, let's take the case where the second building block in magenta was supposed to add an SNAR reaction to this chloride, but maybe that chloride hydrolyzed to give the alcohol here. And maybe it just didn't react. And then we went on to the third cycle of chemistry and a second BBC got incorporated into that position instead of a magenta building block. So if we imagine that this is actually responsible for the binding signal, you can imagine that failed chemistry could have happened for every single monomer along this line. So all of the signal in this line might actually correspond to this unique molecule. So we talked on the last slide about how you can have multiple compounds with all the same DNA code when you have failed chemistry. When you have failed chemistry, you can also have lots of DNA codes that all encode for the same compound as well. So being able to look at these features in detail, understand them by just having experience of interpreting them is very important. Okay, getting to blank adjusted reads. So this is our way of removing the bead signal. So as was mentioned, all Dell screens are going to include some sort of target only, no target control, or something that only has the bead or the matrix. And when you look at those target only or no target control selections, you always see features in that data. So what I'm showing on the left is a with target selection where you see a couple of different line features and kind of a point in space type of feature. And then I'm showing the corresponding blank selection where you clearly see one of those features is present in that as well. When we do analysis, we tend to remove this just simply by using a ratio. So we're penalizing that particular hit if it shows up in the blank. And if it doesn't show up at all, then we use whatever reads were present initially. So when you take the blank adjusted reads now that uses that math, what you see is it cleans up that cube dramatically to only show you those target specific features. So most of our analysis is done with this blank adjusted reads setting, if you will. But of course, we have versions of our tool that can load all of the blanks as well, and you can interrogate that if you would like to using that version. Okay, so this is our Spotfire tool. We call it DEL-DV for DNA-Encoded Library DXP viewer. It actually starts in a blank form, and it's a single Spotfire file for all of our projects, and it's based on a architecture of using on-demand information links in Spotfire. So it's using Spotfire as kind of a query tool to the database where you just load metadata initially, project names and cutoffs and things like that. And then once you make a selection of what data you want to bring back, you hit a button and that data comes back. So we start with our list of campaigns here. You select one of those campaigns and it will populate a bar chart, which is basically a preview of the amount of data that you're going to get back if you load all of that data at a given cutoff. So this is rows of data and cutoffs of reads of 2, 3, 4, 5, 6, 7, 8, 10, 15, 20, 25, etc. We find that loading between 100 and 500,000 rows of data is kind of the sweet spot, but Spotfire is able to handle probably 10, 12 million if necessary, though it's going to slow things down dramatically in particular data loading, and you're just going to get those solid cubes that really don't give you a lot of information. So it's actually pretty quick when you're loading in that 1 to 500,000 row window. Once you decide what cutoff you want to bring back, you put that number in the tool, and then you make a selection of the combination of libraries and selection conditions that you want to bring back. Most of our screens use anywhere from 6 to 10 different selection conditions. So we often are wanting to look at that data at the same time so you can see the compound's response across those selection conditions. So you make a selection of the data you want. You could do this across projects if you want and make cross-project comparisons. And then you hit a button to load the data and it all comes back. And when it comes in, that's when all the magic happens for doing the calculations of line feature strength and diversity. Lots of properties are merged in, so we're not recalculating properties on the same compounds over and over again. And we do pivot tables, so it pivots out by the hit index level, which is the individual molecule, as well as the feature level, so you can do comparison plots of one condition versus another at all those different levels of data loading. When that data comes back, we have a number of different pages that have different workflows populated. The one that most time is spent on is this main line feature analysis page. And you can see the cubic plots in the upper right that have been mentioned a number of different times. You're looking at a view of one library across 8 different selection conditions, which are trellised out for those different selection conditions. And then within those plots, all of the colors are based on the MPO for properties. So the things that have better properties are going to be the yellow-green colors versus red. And then you also see a couple of other colors, magenta and blue, which we'll come back to later, but that's the frequent hitters and bead binders that have been tagged through cross-project analysis in the past in a very similar way that Paul mentioned as well. Of course, Spotfire shows you the max and min reads for the particular visualization, and that will change as you change things. If you make any selection in the top of the tool, you get a details table of all of the relevant data for that selection. And this is, of course, sortable in any way you would like to see the data. It includes monomer structures and properties and identifiers and the MPO score, et cetera. Over on the left, we have a form view of data that allows you to mouse over any point and see all the structures and properties that are associated with that particular compound that your mouse is over. And then on the left is a trellised view of our line feature properties. So we take every point that comes back and we do a calculation to add up the signal in all three directions, X, Y, Z. And then we take the maximum signal that we see for that particular point, and that gets notation of the particular feature it belongs to, and then the feature strength and diversity for that feature. And then we can plot out those features in an XY scatter plot here, where Y is the strength of the feature and X is the diversity of the feature. We can use this as a sorting or selection tool, but also we can track the way those features respond to the different selection condition in terms of their strength and diversity. So here I'm showing a feature that is a diamond, and the shape in this plot is just indicating whether it's a feature in the X, Y, or Z direction. This one is a highly strong feature because it's on the top of this plot, and it's also very diverse to the right of this plot. And you can see it has a very large spot and a couple of small spots going along with that high diversity feature. And then this blue X here is one that's a more uniform feature because it's over to the left of this plot. And you can see it's highlighted and is a very consistent feature. We have a special tool we call Selection Panel that allows you to work with any IDs and select things or filter things based on those IDs. But one of its really nice features is that we can expand a selection very easily. So we can choose a feature in any of these plots or even an individual point, and then use our feature ID in order to say, show me that feature across all of the selection conditions, or filter to that feature across all of selection conditions. That's what I've done here for these two features. I just selected a point in each one of them and hit that button. And now it's showing you that the red feature is present in these two selection conditions, whereas the blue feature is present in all of them except this upper right one. And you can track how those feature strengths change just by tracking the shape of that point and how it moves within those trellised plots. So a very nice tool for just interrogating this data little by little as you're looking at things. And of course, any selection you make will show the detailed monomers down there, so you can do your SAR analysis to see if the things that have the highest signal are giving you similar monomers and good SAR. All of this is linked to all our OnDNA database of anything that we've taken through that OnDNA process, and we have additional information about identity of those binders. So if you happen to pick something that's already been investigated before, it'll show up down at the bottom as well. And of course, this is Spotfire, so possibilities are endless with filters and visualizations for how you can look at this data. Okay, talking about bioprofiles, which is the name we use for describing how a hit responds to selection conditions. A very similar, simple selection graphic is shown here, where you have three different selection conditions, one with a target, one with the active site filled with a binder, and one maybe with a protein partner there. And then you can come up with the response that you would expect over those conditions for various biological profiles. So if it's an active site binder, you'd expect it to be present in this condition, not present in this condition, present in this condition. And the team may then prioritize those different conditions. As I mentioned, we typically do many selection conditions, up to 10 in the experiments. And so these tables can get very complicated. And it's also interesting that often we see profiles in the data that were not anticipated. So they're not what the project team expected. And so we need to circle back with the project team and say, is that of interest to you for a hit that you might want to follow up on? So we've developed a way to kind of pull out these bioprofiles automatically as well. It's shown in a simplified sense down here what the calculation is. And the basic idea is that you need to define your hits in some way. So here I'm just showing 4 different individual hit molecules, and the heat map that you're seeing is the relative reads across the selection conditions, which are in columns. So for this top hit, if we just focus on that one, it did not show up in selection condition 1, 2, or 6, so those squares are black. These are the actual reads in conditions 3, 4, and 5, 972, 341, and 361. And what we visualize is just taking the ratio of the reads to the max reads for that hit. So you get a number between 0 and 1. So 972 divided by 972 is 1, 341 divided by 972.35, etc. And what this does is it takes out the absolute intensity of the reads and really gives you the way, the normalized way for how the hit responds to those selection conditions. So we do that for many, many more hits, and then we can just use the automatic clustering and Spotfire to pull out our bioprofiles. So just showing you that visually. Here's a real project where 20,000 hits are loaded here. We want to take those down to only the ones that we might be interested in. We generally do that by using some sort of a reads cutoff, selecting maybe one hit per line feature so we're not seeing the same things over and over again, and maybe pulling out our frequent hitters, etc. We get that list of things, profiles of interest, and then you can hit a button to do a hierarchical clustering and Spotfire at the row level, and you can really see those biological profiles just fall out of the data. In this case, there was 4 major biological profiles that you can kind of cluster with these magenta boxes. And then if you actually map those to what the project team was looking at, you can use this as a selection tool to interrogate those features. So in this case, the team wanted signal in conditions 1 and 2, and possibly signal in this last condition, but no signal in the middle conditions. And if you look at this map, you can find that profile in this magenta box right here, select those hits, and then you can expand those hits to the features that they're part of, go right to that feature analysis page and really see the profiles of interest. So this is a very interactive process. You can filter this data however you want and do this clustering very easily. And teams really like the visuals that come out of this to see what the actual data is telling them their profiles were, not just what they expected. Okay, so getting to frequent hitters, this is another one that we learned along the way, and it's, again, very similar to what Paul was mentioning. You really need to look across your projects when you're doing this analysis to weed out things that might be leading you in the wrong direction over and over again. And we found this out because almost every project team is interested in what I'll call the allosteric profile, which is something that kind of hits in all your selection conditions. So it's some magical binding site that nobody knows about that we're going to find with DEL screening and give you this great hit. And the problem with that is that, we don't really have a yes, no type of a condition, which often leads to higher confidence in a particular hit. It's just signal that's showing up across the board. The signal is target specific, and I'll illustrate that here with some cubes. So I'm highlighting a number of line features that are all in the same plane, and these features were present for 4 different targets, which are in columns here. And then the corresponding blanks for those no target controls are in the bottom, and you can see those features are not present in the bottom. So these are target specific hits. There's really nice SAR between the lines. There's really nice SAR in the lines. If you look at these in isolation, they really look like real hits. But the problem is they show up again and again and again over these different targets. And in fact, these 4 that I'm showing here actually have showed up in greater than 10 projects to date in our hands. So we call these frequent hitters. They don't, we've done some analysis, and yes, they are promiscuous in the DEL world, but they don't look always like promiscuous compounds. They're not these huge, high molecular weight, greasy compounds that you might expect promiscuous compounds to be. So we can't explain exactly where these are coming from, but they're there, and we know we don't want to take them forward, and we know that they often confirm on DNA, don't confirm off DNA. And that is not something project teams want, because they're putting that resource into making those compounds, and they don't confirm at the end of the day. So what do we do to find these? Well, we summarize across targets at the database level, and then we load that data when the data comes in for those particular hits. And we visualize that rather than filtering it out. And the reason for that is because we've set a threshold for what gets tagged as a hit for a particular project. And if something is just below that threshold in one project, but maybe it showed up for 3 or 4 more, we want it to be tagged. And we also want things within those features not to come through that may be lower level frequent hitters as opposed to the ones that are the strongest ones. So we bring in the data and we visualize it in a different color in our cubes. So, and then as you click on those compounds and look at the detailed data, you'll actually see a list of those targets that those particular compounds hit and how many they were. So you can then make an informed decision on whether or not you want to go forward with a hit that has been seen before. So since implementing this workflow, we're taking significantly fewer weak allosteric profile hits. And this has been particularly impactful for those really tough targets where you don't have tool compounds in hand, you're screening something, you don't really know if there's a binding site or not. It's the long shot type targets because these frequent hitters are usually just above the noise, and that's the kind of stuff that you might select to take forward in those tough targets if you don't have anything better than that. But now we're seeing that if we aggregate all this data across projects, they're showing up again and again. We're not taking anything forward from those projects and saving quite a bit of resource. Okay, just a couple of other capabilities built into this tool. I mentioned we can pivot out at the hit index level or feature level and then plot one condition versus another, and again, use this as an entry point into the feature analysis. So the example shown here is a kinase target, where on the x-axis we have the kinase selection condition alone, the y-axis we have a kinase with an ATP site binder there. So the things that show up only when the ATP site binder is not there are likely ATP site hits, and you can select those and then interrogate that data further. We also have a nice filtering tool that allows you to pick a hit and isolate the signal that is in the planes around that hit with the click of a button. And this is really useful because sometimes you're always focused on that line feature and you see the local SAR within that feature, but there's often lines that are parallel to those lines, and it's really nice to be able to see all of the data associated with any compound that shares a single monomer with the hit that you're interested in. And this makes that very easy to do. And then finally, we've built in a way to look at the negative SAR in monomer space. So this workflow came as a result of getting hits, following up those hits with a project team, and you're sitting in those design meetings, and the questions always come up, what if we change that from a 6-membered ring to a 5-membered ring? What if we put a methyl group there? Was that monomer in the original DEL? And the problem with using your original analysis file is that you're generally only looking at things that were enriched. So you're not seeing the things that should have been there that were not picked up as hits. And there's lots of caveats to using this data because it's primary data. You have all the validation yields that went into these, and et cetera, et cetera. But like Chris mentioned before, this SAR I think is incredibly useful. And we've found that if you have a hit that made it through all the steps and it was the targeted compound, more often than not, you can trust that SAR in the primary data to really help you design your follow-up steps. So the way this tool works is you choose a cube and then you just choose the cycle that you want to look at, the particular monomer cycle, and it will load a table that contains all of the monomers that went into that particular library for that cycle, including structures. And then when that comes in, it'll merge in the max signal for that particular monomer that was observed. This is all linked up to the cube. So if you click on one of these rows, it'll highlight the plane associated with that particular monomer and show you all the data in the details table. But what's really great here is you can do sorting by similarity. Here I've done a similarity search on the highest reads compound, and I'm showing other compounds with very high similarity to that particular monomer, and then the reads that were observed in the actual screen. And these down here in red were ones that are high similarity that are not in the data set because they didn't show up at all. So this is a really nice way to look at that negative SAR and not go and make things that you're pretty sure are not going to be active based on that primary data. Okay, so just one slide on the overall process of how we use these tools. Of course, before the screen, it's very important to work with the team to really understand the goals, priorities, the target interacting partners, any chemical matter that is there, the need for binders versus functional lead matter, and summarize that into a nice little slide deck that gives you all that information that'll help you during triage. We then develop our selection conditions and define the expected bioprofiles with a table similar to the one I showed before, but usually much more complex. We also like to define a primary and comparator selection conditions. And this has come from the fact that during selection, we've talked about sometimes there's a need for doing different rounds of selections based on the amount of DEL that is retained after each round. And we found that if you really want to compare two samples together, if you do different number of rounds of selections for those conditions or those selection conditions, you're not going to be able to make a good comparison there. So even if one of them is way out of spec with regard to how much is retained, we like to do equal rounds of selections for the samples we're directly comparing. And then we'll just sequence more or less of a sample if necessary in order to fit the right amount of material onto those chips. Once the data is in the database, we use our tool to take a very high level look at the data. And this is usually done with the data where the blank is present, not the blank adjusted reads. Here we evaluate the need for normalization across conditions. If you're looking at, a selectivity target, do you see all the bead binders kind of on that y equals x line, or are they 5-fold, 10-fold off? Because that might impact how you interpret selectivity in the selection for the hits that you really think might be selective. We also always evaluate the blank overlap with the selection conditions in a plot like this. So this is showing the selection conditions in bar chart and just adding up the total signal that we're seeing for each selection condition. And then I'm coloring it based on whether or not those hits also appear in the blank or not. And this really gets, if you see a plot like this and it's mostly red, and we have seen that where 99% of the signal that we're seeing was also present in the no target control, then that goes to the need to get rid of those bead binders in some way, either through a bead depletion step or pre-clearing your library by subjecting the library to the beads before you do the selection. So this is a nice overall look at how much useful information is coming out of your sequencing. Then we load our data with the adjusted reads, and we evaluate replicates and noise level and determine cutoffs, things like that. Look at signal distribution across libraries, make sure we're seeing kind of what we expect there. And then all of this bioprofile clustering can be done as an entry point into finding features. And then the real feature analysis is what takes the most time. And I don't like to automate this. I like to just take a couple of days. Our libraries aren't numerous enough that we can't just look at all of them little by little, but also start from the bioprofile clusters, start from pivot plots, use MPO filters, etc. Just go through and find all the hits that may be of interest to a project team and try to characterize those things I talked about, whether the hit has good primary SAR, whether it has good properties, how it relates in terms of the biological profile, and collect a list of those hits that'll later be prioritized. Of course, if you find those bioprofiles or other unexpected findings, that's a great time to circle back with the project team and see if there's interest in those other things. And then finally, a step that is always necessary is a prioritization. We all have limited resources. The team's only going to take forward a certain amount of things. You want to give them options, but you also don't want to just throw them 50 things and say, you choose from these, because we're the experts on looking at this DEL data and what it means. So we always prioritize kind of from high to low, all of our hit proposals based on the pieces of the puzzle that I talked about, the raw signal, the SAR we see in the data, the properties, and the coverage of those bioprofiles. And then we collect that into a tool that we use called PCAD, it's called, for the Pfizer compound analysis tool. This is just a structure viewer, like an SDF file viewer, that will be one row per structure with all the properties and bioprofiles of those compounds. And that'll allow the team to very easily run their own computational models, et cetera, on those hits and make a decision on which ones they want to take forward. So flexibility is the key to this whole process. Having this flexible analysis tool based in Spotfire, knowledge of all these things leads to the best outcome in the end. So final slide, talking about success a little bit. So platform success for us is finding a binder of interest with off-DNA binding less than 20 micromolar. That's what we set out as our metric from the very beginning of this endeavor as what is a hit. But that's not real success. Real success is making that hit interesting enough for that project team to engage their own chemistry resource toward optimization and hopefully a lead series, stage gate transitions, and of course a drug at the end of the day. After those hits are found, you don't want to just throw it over the wall and let the project team take over because of all that great primary data that you have. So participate in chemistry meetings to guide that initial design, get the most out of that primary SAR from the screen, use both the positive and the negative primary SAR. And I tell teams to trust it, but verify it. So if there's a close-in compound where you want to put a methyl group and the DEL tells you it's not a good idea, but it's really important to know for sure if it's a good idea or not, make that compound. But if it's something that you're going to do a whole nother library where you use the exact same disconnection that was done in the DEL and you've already investigated that with 5,000 monomers, I would not redo that library unless you're going to change it to different monomers, et cetera. So it's best to change things that weren't covered in the DEL, use alternate disconnections. If it's in a templated library, modify that template, and always investigate that linker connection SAR because we've seen some cases where it's very important to the activity that you see. And just as I was communicating some of these design ideas to our project teams internally, this great paper came out from Janssen that talks about trends in hit-to-lead optimization following DNA-encoded library screens, and it's very much parallels a lot of things I'm saying here. So I think this is a good set of rules to follow. So bottom line, partner with those project teams long after the DEL screen triage to get the most value out of your screen. With that, I'll stop and thank a number of people. Great team at Pfizer in collaboration with HitGen for this entire process. Really, really fantastic to work with all these people. And I thank you very much for your attention. And I'd be happy to answer any questions.
Great, thanks so much, Justin. I like that you figured out a way to incorporate the negative primary SAR into the visualization. It comes up all the time, like you said. It's nice that you have an easy way to look at that. Okay, so I guess I'll start with the one at the bottom here. Do you account for known chemical matter when analyzing the cubes?
So if you mean known chemical matter of overlap with file compounds, there's very little overlap with our file. The DEL is really different chemical space, I would say, than the rest of our file. If you're talking about within other projects that have worked on DEL, then anything that has gone through the process of on DNA synthesis and confirmation, all that data will be present while you're doing the analysis. So you can see it.
Okay. Justin, do you also take into account the performance of synthons when validated against the model substrate pre-library production when looking at the selection data?
Synthons when read that one more time.
Yeah. Do you also take into account the performance of synthons when validated against the model substrate pre-library production when looking at the selection data in addition to analogs?
I'm not sure I understand exactly what's being asked there. So
anonymous attendee. So maybe, I don't know if there's a way to like type the answer and chat about it or something.
I'll be happy to answer in chat.
Okay, maybe they'll chime in again. Okay, let's see. There's another one. Any possibility that some of your frequent hitters are reversible covalent?
I doubt they're covalents. Just because they don't have the functionality you expect in a covalent inhibitor. If I had to guess, it's, maybe there's common protein folds that somewhat recognize some of these hits. And I don't think they're strong interactions. And I think the linker and the DNA piece may contribute to those interactions as well. So I think it's kind of like a collection of weaker interactions, which is leading to what we're seeing. And that's why when we make them off DNA, we kind of lose that. It might also be something to do with just surface bound proteins. So we sometimes do on DNA type SPR experiments, and we'll see those same hits show up in those types of experiments as well, which is another surface binding type assay, as opposed to something that's in solution or a functional assay where we're not seeing them as hits. So speculation at this point. I don't have a great answer, but those are my best guesses right now.
I guess related to that about the frequent hitters, someone asked if it might be tag specific. Do you have any
So
on
we
that, I guess.
certainly see some across tags.
Yeah.
Certainly bead binders are very, are tag specific because the matrix changes at that point, but we do see the frequent hitters across tags.
Okay. And then I know we want to make sure we have some time for a break, so I'll go to the top ranked question here. It says, "Hi Justin, can you share the slide you mentioned about how on DNA resynthesis outcomes track with your libraries?"
Sure.
Maybe this, yeah, I guess you know what I'm asking. So
yeah, so this is kind of the stats on our on DNA process. So the first one's around library fidelity. I lost my pointer here. So you'll have to believe what I'm pointing at. So we've done 290 analogs on DNA. This is only hits, so it's not really a random representation of our libraries. And then the targeted compound is present at these yield cutoffs at the various levels. So if you take the middle pie, 25% yield or more, 60% of our library fits into that box. With the down to 5%, it's much higher than that. When we take that to binding, so binders versus the targeted compounds, if you focus on the pie on the right there, these are examples where we found the binder. 74% of the time, it is the targeted compound. And finally, off DNA confirmation of the ones where we have a complete data package, we're running at about 45% where we find that binder of less than 20 micromolar for the examples we have. I will note this is highly project dependent. Some projects do well, some projects do worse. And a lot of our projects are what many would consider to be very tough targets where no other technologies have worked to find hits. So we throw DEL at it to see what happens. This number is also a little bit low because frequent hitters, were incorporated more recently. And a lot of these red ones here, we've now deemed as frequent hitters that we would not have followed up on in the future. But the key point to make about the whole platform is this whole red pie, we're not making anymore. And then all of this stuff, we're not making anymore. So the number of compounds we're making off DNA is much, much less with this improved data package.
Great. And I guess we're running short on time, but one last question.
Sure.
Do you have a reliable way to rank the affinity of the hits before off DNA synthesis?
Before off DNA synthesis, with the Bally mass spec platform, we do get a ratio of an enrichment ratio, basically. So that is a good way to rank the affinity of the hits because it's taking into account the ratio of the target selection versus the bead only selection for that one component of that mixture. And that does seem to correlate well with the eventual binding.
Okay. So there's a few more questions. I'll let you answer them if you'd like in the chat. And I just want to thank everyone for joining us so far today. We have a short break and we'll return at 3 p.m. This is when I wish we were live and in person since I see a lot of familiar names on the list. I wish I could say hello to folks, but feel free to go grab a cup of coffee and come back at 3 for a talk on case studies and hit to lead optimization for DEL derived compounds.